Inheritance is the transmission of traits from one generation to the next. Genes are made of DNA and they are passed through gametes. Gametes are a product of meiosis, but how does meiosis work? Meiosis consists of two consecutive divisions, meiosis 1 and meiosis 2. Meiosis 1 has prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. In prophase 1, homologous chromosomes pair up and exchange genetic material by crossing over. In metaphase 1, homologous chromosome pairs align randomly at the metaphase plate using independent assortment. In anaphase 1, homologous chromosomes separate and move to opposite poles. In telophase 1 and cytokinesis, two haploid cells form, each with half the original amount of chromosomes. In meiosis 2, you have prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. In prophase 2, new spindle fibers form around the chromosomes. In metaphase 2, chromosomes align at the center. In anaphase 2, sister chromatids separate and move to opposite poles. In telophase 2 and cytokinesis, 4 haploid gametes form, each genetically unique. Meiosis is important because it maintains genetic stability across generations and increases genetic diversity. Let's talk about Mendelian genetics now. Mendel began by choosing a simple, obvious trait to study: flower color. He noticed that the flowers of some plants were purple, while others were white. These traits, purple and white, were variations of the character flower color. In biology, a character is a heritable feature that can vary, like flower color. The different versions of a character, like purple or white flowers, are called traits. He also noticed that pea plants could be either true breeding, meaning that if they were allowed to self-pollinate, they would always produce offspring with the same trait, or not. True breeding plants would always produce offspring with the same flower color when self-pollinated. Mendel decided to cross two different true breeding pea plants. One had purple flowers and the other had white flowers. This process of mating two contrasting varieties is known as hybridization. The parent plants were called the P generation, short for parental generation. When the two plants crossed, Mendel observed that all the offspring, the F1 generation, had purple flowers, but Mendel was confused. According to the popular blending hypothesis of inheritance at the time, he expected to see a mix of purple and white flowers in the F1 generation. Instead, every single one of them was purple. Mendel didn't just accept this result, he delved deeper into the mystery. What he discovered changed the course of science forever. When he allowed the F1 plants to self-pollinate, something extraordinary happened in the F2 generation. Some of the plants had purple flowers, while some had white flowers. The ratio was about 3 purple flowers to 1 white flower. Mendel realized that flower color didn't blend in the F1 generation. Instead, the purple trait was dominant over the white trait. The white flower color didn't disappear, it just seemed to be hidden in the first generation. Mendel concluded that each plant must carry 2 factors, which we now call genes, for each trait, one inherited from each parent. This discovery became known as the law of segregation, which states that the 2 alleles for a character segregate during the formation of gametes. This means that each gamete carries only 1 allele for each gene. When the F1 plants produced gametes, half carried the purple allele and half carried the white allele. Mendel called the 2 different versions of the flower color genes alleles. The purple flower allele was dominant, meaning that it would always express itself if it was present. The white flower allele was recessive, meaning it would only show up if the plant inherited 2 copies of the white allele, one from each parent. Mendel used Punnett squares, simple charts to predict the probabilities of offspring inheriting certain traits. These squares help Mendel visualize how alleles combine during fertilization, providing a tool to predict genetic outcomes. As Mendel continued to study other traits, he realized that there was a difference between an organism's genotype and its phenotype. The genotype refers to the genetic makeup of an organism, the combination of alleles it carries. For example, a plant might have the genotype homozygous dominant or heterozygous for flower color, both resulting in purple flowers. The phenotype refers to the physical appearance of an organism, which is influenced by its genotype. For example, both a plant with the genotype homozygous dominant and one with heterozygous would have the same phenotype, purple flowers, because the dominant purple allele determines the flower color. Mendel wasn't satisfied with just studying one trait. He wanted to know if there was a pattern when plants inherited 2 different traits at the same time. He decided to cross 2 plants that differed in both flower color and seed shape. The plants were dihybrids, meaning that they were heterozygous for both traits. Mendel's second law, the law of independent assortment, came from this experiment. It states that the alleles for different traits segregate independently of one another during gamete formation. This means that the inheritance of one trait does not affect the inheritance of another trait. Through this, Mendel saw that the inheritance of flower color and seed shape followed predictable patterns, much like the law of segregation had predicted for single traits. Mendel's experiments also helped explain various patterns of inheritance. For example, complete dominance occurs when one allele completely masks the expression of another, like the purple flower allele masking the white one. But Mendel also knew that sometimes things weren't so simple. In incomplete dominance, the heterozygote shows a blend of both traits. For example, in snapdragons, crossing a red flower with a white flower produces pink flowers in the F1 generation. In codominance, both alleles contribute equally to the phenotype. A famous example of codominance is seen in human blood types, where both the A and B alleles are expressed in individuals with type AB blood. Before we go over how the movement of chromosomes during meiosis explains Mendel's law of segregation of alleles and Mendel's law of independent assortment, let's compare and contrast mitosis and meiosis. Mitosis produces identical diploid cells, while meiosis creates unique haploid gametes. Mitosis has 1 division, but meiosis has 2, meiosis 1 and meiosis 2. Genetic variation occurs in meiosis through crossing over and independent assortment, while mitosis maintains genetic consistency. Both start with a diploid parent cell. Both involve chromosome replication before division. Both followed the same basic stages: prophase, metaphase, anaphase, and telophase, but meiosis has 2 rounds. So anyways, Mendel's law of segregation explains that alleles separate during meiosis, so each gamete gets only 1 allele from each parent. For example, if a pea plant has 1 allele for purple flowers and 1 allele for white flowers, these alleles will separate when making gametes. Some gametes will get the allele for purple flowers, and some will get the allele for white flowers. This is why offspring inherit 1 allele from each parent. Mendel's law of independent assortment explains how different genes sort separately. For example, a plant with one allele for purple flowers and one for white flowers, and one allele for tall height and one for short height, will pass on the flower color alleles separately from the height alleles. This means a tall plant with purple flowers could have offspring that are short with purple flowers or tall with white flowers, depending on how the alleles combine. Chromosomes control this because they carry genes. During meiosis, homologous chromosomes separate, following Mendel's law of segregation. Later, different chromosome pairs line up randomly, following the law of independent assortment. That's why traits are inherited independently and why siblings can look so different. Let's learn how to do chi-square goodness of fit test. Chi-square goodness of fit tests are used to determine if there's a significant relationship between two groups of data. Observed outcomes are compared to expected outcomes to see if variations could be due to chance. Often used to test genetic crosses, can also be used for other analyses. To perform a chi-square goodness of fit test, you must first establish the research question, then you must determine the null hypothesis and the alternative hypothesis, then you want to count the observed values, then you want to determine the expected values, then you want to calculate the chi-square value, then calculate the degrees of freedom, and then select the p-value, and then finally identify the critical value. A karyotype is a visual representation of an individual's chromosomes arranged in pairs from largest to smallest. It is used to detect chromosomal abnormalities including aneuploidies, which occur when there is an abnormal number of chromosomes. To interpret a karyotype, one must first count the total number of chromosomes, which should be 46 in a typical human. Any extra or missing chromosomes indicate aneuploidy, such as trisomy 21, also known as Down syndrome, where there is an extra chromosome 21, or Turner syndrome, where a female has only one X chromosome instead of two. Structural abnormalities like deletions or translocations can also be identified. Aneuploidy is primarily caused by nondisjunction, an error that occurs during meiosis when chromosomes fail to separate properly in anaphase 1 or 2. This results in gametes with either extra or missing chromosomes, which, if involved in fertilization, lead to disorders like trisomy 18, Edwards syndrome, or Klinefelter syndrome, XXY in males. Since the environment does not influence aneuploidy, it is purely a result of errors in cell division. Sex-linked inheritance refers to the transmission of genes located on the sex chromosomes, primarily the X chromosome and in rare cases the Y chromosome. Since males have one X and one Y chromosome while females have two X chromosomes, the inheritance of sex-linked traits differs between sexes. X-linked traits are more commonly expressed in males because they have only one X chromosome, meaning a single recessive allele on the X will result in the trait being expressed. In contrast, females who have two X chromosomes must inherit two copies of a recessive allele to show the trait, while those with only one copy are considered carriers. Examples of X-linked disorders include color blindness, hemophilia, and Duchenne muscular dystrophy. Y-linked traits, on the other hand, are extremely rare and only affect males, as the Y chromosome is inherited solely from father to son. In X-linked recessive inheritance, a mother carrier has a 50% chance of passing the affected genes to her sons, making them affected, while her daughters have a 50% chance of becoming carriers without showing symptoms. If an affected father mates with an unaffected mother, all sons will be unaffected, while all daughters will be carriers. This pattern of inheritance explains why genetic conditions are more common in males and highlights the significance of the sex chromosome in genetic variation. The environment plays a significant role in shaping an organism's phenotype, even when the genotype remains the same. Various environmental factors such as temperature, nutrition, sunlight, stress, and chemical exposure can influence gene expression and lead to differences in physical traits or behaviors. For example, in certain reptiles, the temperature at which eggs are incubated determines the sex of the offspring, while in humans, poor nutrition during childhood can stunt growth despite genetic potential for tall stature. Sun exposure can also cause an increase in melanin production, leading to darker skin, while stress and lifestyle choices can impact metabolism, height, and overall health. Additionally, exposure to toxins or pollutants can trigger changes in gene expression, sometimes leading to disease. This ability of organisms to develop different traits based on environmental influences is known as phenotypic plasticity, demonstrating how genes and the environment work together to shape an individual. Let's identify different pedigrees. In an autosomal dominant pedigree, the condition appears in every generation. Both males and females are equally likely to be affected. Affected individuals have at least one affected parent. This means you won't see the condition skipping generations. If a parent has the condition, there's a 50% chance they will pass it on to their offspring. In the pedigree, you'll see squares, males, and circles, females, shaded in nearly every generation, indicating that the trait is continually passed down. An autosomal recessive pedigree will show the condition appearing less frequently and can skip generations. Both males and females are equally affected. Affected individuals can be born to parents who are both carriers, unaffected themselves. This leads to patterns where siblings might be affected without either parent showing symptoms. Carriers are typically denoted by a half-shaded symbol, though often only the affected individuals are highlighted. It's common to see the condition show up in siblings, while previous and subsequent generations are unaffected. In an X-linked recessive pedigree, the condition primarily affects males, as they have only one X chromosome. Females can be carriers without showing symptoms because they have a second X chromosome that compensates. The condition tends to skip generations, as carrier mothers can pass the trait to their sons. Affected males cannot pass the condition to their sons, but their daughters will be carriers. In the pedigree, more males, squares, will be shaded, and fewer females will be affected, but some might be carriers. An X-linked dominant pedigree is characterized by affected individuals in every generation. Both males and females can be affected, but the pattern is slightly different than autosomal dominance. Affected males will pass the condition to all their daughters, but none of their sons. Affected females have a 50% chance of passing the trait to both sons and daughters. In the pedigree, you will see both squares and circles shaded in every generation, but with a noticeable pattern of affected daughters from affected fathers. Using probability to analyze and predict the passage of genes from parents to offspring involves applying the principles of Mendelian genetics, primarily the laws of segregation and independent assortment. When considering a monohybrid cross, such as crossing two heterozygous individuals, the offspring's genotype can be predicted using a Punnett square. The probability of producing offspring with the genotypes homozygous dominant is 25%, heterozygous dominant is 50%, and homozygous recessive is 25% respectively. In more complex scenarios, like a dihybrid cross, you would calculate the probability for each trait separately and then combine them using the multiplication rule. For example, if you cross two heterozygous individuals, the probability of the offspring having that genotype is determined by multiplying the probabilities of each trait, which would be around 1/4. By employing the multiplication and addition rules of probability, you can accurately predict the likelihood of different genotypes in offspring, enabling a deeper understanding of genetic inheritance patterns.